Hi everyone, welcome again to the course of structural biology. For last few modules we have discussed about the core of the course, the structural biology techniques. Now we are out of that. In this module we will talk about visualization. So, to start visualization, very interestingly I will take you to the first slide which was used to start the course. Why we should learn structural biology, and one of the reason I started with is seeing is believing. I also have quoted Kenneth Turnbull, who told a picture is worth 1,000 words. Today I am going to talk about a module which is about visualization. But interestingly, when I started I talked about the picture visualization. Now it is not limited to the visualization of the picture. Through the course I have shown a lot of movies. So first of all, the techniques have improved a lot, the computational power improved and all. So today I will talk about visualization, but the whole module include the history of molecular visualization, because it is very interesting for you to know about how actually the visualization happened, from where it started, from where people have started the idea of making it under computation, what they did before computation, all would be under history of molecular visualization. Then come process details of handling PDB and MTG files. In the last modules we have told about the structure is deposited under PDB, and MTG are where the electron density you will get. So for a X-ray crystallographic structure, for a cryo-electron microscopy you need a electron density map. We will talk about them. Then we will talk about the principle behind visualization, what are the type of analysis we do and what they are having impact on the application. And then we will discuss popular softwares, the popular softwares like Qt, Pymol. Why Qt and Pymol, I will describe that also. So let us welcome to the history of molecular visualization of biological macromolecules. So as I talked, why do we need visualization? Since learning is abstract, especially when dealing with what we cannot see. We have talked about the power of an eye. So what is not coming under the power of an eye? We need some instrumentation, starting from a simple lens, go to compound microscope, electron microscope, getting more powerful resolution, other spectroscopic signals which will give us information, all those comes under this. If we could see that, we could image them in microscopy. If we cannot see them, we take the information and build up the models. Computer modeling has provided a window to the unseen and transformed science research. So before, a lot of things people have to imagine themselves, but now with the advent of high resolution microscopy, spectroscopy, computation, we could really see the unseens. The gap between the research lab and the science classroom is getting smaller. So one of the thing was what the scientists are doing, what they really see. Can we, the students, could see them? It was not possible. Sometimes it was very expensive, the setups, the instruments. So how the journey happened is the topic of today. Before computational visualization or computer visualization, softwares were developed, molecular structure were presented by physical models of metal wires, rods, spheres. So people see the signals and from there they want to develop a real life model. With the development of computer hardware and software technology and computer graphics, programs were developed to visualizing and manipulating three-dimensional structure. The computer graphics help to analyze and compare protein structures to gain the functions of protein. It is applicable to all biological macromolecules, but as I always say, protein is the functional molecule, so a lot of interest are always there in focus to the protein. Molecular visualization helps the scientists to biomedical experts to study, understand and engineer the biological macromolecules. So, as I told in the previous module, structural information is vital, but able to look at in high resolution is always helping our imagination power to be used in higher level. And from understanding to a system, understanding a protein, a DNA and all, to engineer them comes from that concept. When we see them, when we compare between them, when we compare between, as we say, when we are imagining, the comparison was difficult, but when we are comparing, initially we are comparing between a, let us say, a apple with an pineapple, then we are comparing between apple and orange, and now we are getting opportunity to compare between apple and apple. That is where we are fortunate, we could see them, we could compare them in front of us, and from there, from understanding of the real system, because we have now very good understanding of the real system, we are going to engineer them. A brief of that I will talk about in the protein engineering part of the course. User friendly graphic interface makes the area of bioinformatics a fulfilled scientific thrill to bioscientists. For the first X-ray crystallographic solution of a macromolecule, we all know myoglobin, Kendrew and co-worker built brass model at a scale of 5 centimeter per angstrom. This is the brass model. The models were built and supported within 2500 vertical rods arranged to fill a cube 6 feet, that means 2 meter on a side. The colored clips were attached to the rods to signify electron density and guide the building of the model. The forest of rods obscured the view of the model and made it hard to adjust. It is much difficult than what we do nowadays, a computer model. Its size made it cumbersome and problematic to move. So, it stayed in one place because it is heavy, you cannot take it in the class. So, as I start with a difference between where the work is going on or where the model is established from where the students are coming, there was a gap. The Richards box, aka Fred's folly. In the late 1960s, while solving the structure of ribonuclease, Fred Richards and co-workers introduced an optical comparator that facilitated building a Kendrew style brass model. Electron densities resulting from crystallographic solutions were printed by computer on paper. This is the first time when computer actually is coming to be involved, and from there actually the idea of computer graphics are developed. And electron density contour lines were traced by connecting numbers of similar values on the paper. These contour lines were then traced onto transparent plates 3 into 3 feet. The plates were mounted vertically equally spaced, creating a sliced 3-dimensional electron density map. Half silver mirrors were arranged to superimpose the electron density map upon the brass model. As larger molecules were solved, the scale was reduced to 2.5 centimeter per angstrom and then 1 centimeter per angstrom. So, there is a even journey to make finer structures. Several Kendrew style models built by Michael Rossman's group, including one with a still assembled Richards box, are on display in the basement of Lilly Hall at Purdue University, West Lafayette in US. So, you all know about Michael Rossman. Michael Rossman have developed molecular replacement, contributing greatly to the advancement of X-ray crystallography. So, in his group they have developed a lot of models, and there's like some of them are still there. A Kendrew style model is on display at the US National Institute of Health Museum, Bethesda, US. This model, built with a Richards box, represent the Fab fragment of an antibody built by Eduardo Padlan in 1974. A Kendrew style model of chymotrypsin built by Alexander Tulinsky is on exhibit in the main lobby of the chemistry building of Michigan State University, East Lansing, Michigan. Getting it there from the room where it was originally built required partial disassembly because it would not fit through the door. From all those initial level model coming to physical ball and spoke model, where from the macro arrangement of the helicity, atomic level representation is introduced. After Kendrew et al. Solved the structure of myoglobin, they built a physical ball and spoke model. Eric Francoeur had provided an illustrated account of this era, which is A. Barker's model of myoglobin, which you could see. 29 of these models were sold to researchers around the world in 1960s, manufactured by Beaver's miniature models, and outfit still offering a variety of protein models today, including custom proteins. So, the company is still there, but that time making them contributing to science, because scientists were actually looking at that and design further experiments. So, it was very important. Now, it is not so important because thanks to the improvement of computational modeling, but still for educational purpose, to make people interested in protein related structure related work, these models are indispensable. Bryon's bender, Byran Rubin, while working as a crystallographer with Jane Richardson in the early 1970s, invented a machine of bending wire to follow the backbone trace of the protein. In 1970s, computer visualization of macromolecules was not yet generally available to crystallographers. Kendrew style models were built on wire supports from projections of the electron density map from isocontours traced on plates of glass in a Richards box. These models were large and cumbersome. The small backbone wire models from Byron's bender were the most manipulable and portable models available at that time. So, as I told when I was talking about the previous models are made of metal and all, so they were very heavy, very difficult to carry and all. Byron's bender makes a revolution in comparison to that. An example illustrating the importance of models from Byron's bender occurred at scientific meeting in the mid 1970s. At this time, less than 2 dozen protein structure had been solved, as we have already looked at while our journey in structural biology. David Davis brought a bender model of an immunoglobulin Fab fragment, and Jane and David Richardson brought a bender model of superoxide dismutase. While comparing these physical models at the meeting, they realized that both proteins use a similar fold despite having only about 9% sequence identity. So, at that time, as I told, the computational analysis, the computational alignment, sequence alignment, structure comparison was not available. So, David Davis group, they have solved the immunoglobulin structure. David Richardson group, they have solved the superoxide dismutase. But when both the structures were given to them, the Byron's bender, they have developed the models. You look at the physical model and you realize them, these two models are very similar. So, the folds are similar, though the sequence is very dissimilar, only 9% sequence identity is available. So, this is not only helping you to learn, this is helping you to understand science. This is one of the major breakthrough where understanding about the sequence to fold. There is always thought about the correlation between sequence. If there is high identity, there is a chance of having similar fold, but it could be otherwise also. Similar fold could be found without having sequence identity. Byron's bender have help scientists looking at that physically get a view and understand. This incident was the first recognition of the occurrence of what is now recognized as the immunoglobulin superfamily domain in proteins that are apparently unrelated by sequence. The insight was published in a paper entitled Similarity of Three-Dimensional Structure between the Immunoglobulin Domain and the Copper Zinc Superoxide Dismutase Subunit. Byran's bender remained available through the 1990s. Tim Harman of the Medical College of Wisconsin was one of the last avid user of the models. Tim brought the bender into the local high schools and taught teachers and groups of students how to construct model. Harman's website has photos of many beautiful wire models as well as many more of the history, technology and source of Byran's bender. So, if you are interested, go to Harman's website and take a look. Aside from the importance of the tactile as well as visual input these models provide, another of their great strength is that they jiggle and vibrate when fondled, thereby simulated thermal motion. So, I just try to help you understanding that. So, those are light wares, right? Light means lightweight wares. So, depending on how the side chains are, they respond to a force. You understand? So, suppose this is one branch and that is other. If this is having bigger amino acids, this is having lighter or smaller amino acids, they would like jiggle or they would vibrate differently, giving idea about the thermal motion. Too often users of computer models lose sight of the fact that protein molecules in living system are constantly flexing due to thermal motion, but these models help you understanding them physically. Now, coming to molecular sculpture, shortly after convincing the idea from his bender, crystallographer Byran Rubin realized that the machine used in the Midas muffler shops to customize automobile tailpipes operated on a similar principle, but at larger scale. He collaborated with the local shop to construct a backbone sculpture of rubredoxin about 5 feet high from stainless steel tailpipe. So, this was used in the other purpose automobile tailpipes, and Byran Rubin tried to use them in developing model and develop the model of rubredoxin. Rubin's rubredoxin sculpture won the Chandler competition at the University of North Carolina in 1973 and since then has stood in the lobby of the Paul M. Gross Chemistry Building at Duke University, Durham, US. In the mid 1990s, Rubin resumed the creation of molecular sculptures such as collagenous ribbon model shown. Larger sculptures are under construction in Rubin's workshop near Rochester, New York in 2000. Other molecular sculptures made by Bethesda Grossman renders protein within glass blocks. He makes laser crystals in which the macromolecule DNA double helix, as you see here, is represented by tiny laser induced fractures within a glass block. If you search Bethesda Grossman, you will see a lot of such laser models. They are still working, a lot of people, lot of crystallographers, electron microscopists, they used to gift their students or people they want by making those models till now. Julian Vos Andrea makes large protein sculpture in metal and other media. So, this is a model of mating pheromone. Edgar Meyer makes molecular sculpture in wood using a computer controlled milling machine. So, this is the model made by Edgar Meyer. Kenneth Eward offers a gallery of virtual sculpture and photography, but somehow they are not available nowadays, but they contribute that time significantly. Rapid prototyping. In the late 1990s, efforts were begun independently by Michael Bailey, San Diego Supercomputer Center, and Tim Harman, Milwaukee School of Engineering, to use the new engineering technologies for rapid prototyping for building physical models of the proteins. Custom models of research quality became available in 2000 from 3D molecular designs. So, this is one they produced, which is light harvesting complex 2, built at the SDSC telemanufacturing facility and painted by hand according to the actual content. This is ATPase, a molecular motor built at the MSOE Center of Biomolecular Modeling and again painted by hand. Coming to the computer representations. So, these what you are looking at, these are early representations which are later brought together and made movies. So, as early as 1964, Cyrus Leventhal and his colleagues at MIT had developed a system that displayed on an oscilloscope rotating wireframe representation of macromolecular structures. Online excerpts from 16 millimeter movies of these early computer renderings have recently been made available. As I told, for more on computer rendering, you could see Eric Frankio's gallery, you could get many of them, but these are example how the initial attempt to make structural visualization, structural graphics was done. Then come ORTEP. In 1965, Carol K. Johnson of Oak Ridge National Laboratory released ORTEP, a program to produce stereoscopic drawings of molecular and crystal structure with a pen plotter. So, it is a pen plotter which is you could use in the for plotting graphs and all, but this was actually very effective. I am coming into. Created in Fortran and first released in 1965, ORTEP, Oak Ridge Thermal Ellipsoid Plot Program, rapidly became a favorite of small molecule and protein crystallographers to produce illustration of structures for conference presentation and publications. A key strength to ORTEP was its capacity to generate stereoscopic images automatically. So, as I told, the stereospecificity, the chiral environment is a real interesting thing for protein. So, because ORTEP automatically generates stereoscopic images, it got instant popularity. ORTEP 2 was released in 1976 and 3 was released in 1996. In 1966, C. K. Johnson produced the stereoscopic drawing of myoglobin, vitamin B12 coenzyme and poly L lysine for presentation at the Second International Biophysics Congress in Vienna, Austria. A reproduction of these drawings gives a good indication of ORTEP's capabilities. This was a time when protein crystallographers was just becoming able to determine the structure of proteins, and they clearly appreciated that ORTEP could facilitate the interpretation and publication of their results. So, this is the stereoscopic illustration of the alpha helix produced with the ORTEP. One of the very good example of how the crystallography community of that time was excited comes from a letter showing John Kendrew's reaction to the drawings of the myoglobin produced by Johnson. So, this is the letter. I am not going into details. The two lines indicates, dear Dr. Johnson, I have just had a letter from Herman Watson enclosing your extremely beautiful pictures of myoglobin. I am quite delighted by them and do congratulate you on the quality of the results. So, that will help you to understand a letter from Kendrew, that time Nobel laureate, how excited he was, and this is the original letter coming from Kendrew. In the mid 1970s, a protein structure was solved crystallographically and visualized entirely with computers for the first time without building a physical Kendrew model by David and Jen Richardson and colleagues. I have given the paper so that if you are interested, you could go to the paper and go to the details of the work. They used a density fitting computer system called GRIP at the University of North Carolina. In the late 1970s, more and more crystallographers made the transition to building models for newly solved protein crystals with computers, the electronic research boxes rather than physical Kendrew style model. One of the large advantages was that the computer kept track of the atomic coordinates, whereas atomic coordinates had to be measured manually from a Kendrew model atom by atom. You have to actually physically measure and then develop or construct a physical model, but for a computer, if you could construct, if you could know the conversion from the atomic information to the model, computer could do that by itself. So, that is the major advantage. A history of electronic research boxes and the 4 computer systems most widely used at the time is available in Michael J. Connolly's Molecular Surfaces, a review in the computational chemistry section of the Networks website. That is a really informative and revolutionary review, because what Connolly have worked on molecular surface, if you see all the current visualization programs, the servers, they all use that principles. In 1977, a Warframe Atlas of Macromolecular Structure was published. Also in the late 1970s, Thomas K. Potter, then of the Division of Computer Research and Technology, DCRT, NIH, USA, developed computer algorithm for shaded space field representations. These developments revolutionized the visualization of macromolecules, but were available to a limited number of specialists with access to the most powerful computers at the time. So, they are specific for a specialized setup of computer which are very expensive. So, even the common scientists cannot get access to them. And then come TAMS, which is called Teaching Aids for Macromolecular Structure. That is the one of the initiative come from DCRT. DCRT found it to be prohibitively expensive to publish an atlas of computer generated space field images in color. A breakthrough occurred when they learned of an inexpensive cardboard viewer of stereo slides. You could see here in stereo slides, and you could get the stereo specific view, which would accommodate a pair of 35 millimeter slides in 2 into 2 inches mounts. This viewer folded flat of convenient enclosure in the cover pocket of a 3-ring binder in which TAMS was distributed. So, even when you get the publication, you get the journal, you get the viewer in package. So, you could see the protein, you could get the stereo specific view. Satisfactory stereo projection was obtained by using 2 conventional slide projectors with polarizing filters, each viewer wearing polarized glasses. This is how it could look, the human immunoglobulin G. As you could look at the stereo specific or stereoscopic views, you will see the beauty beyond any previous experience. So, that was the revolution. We feel that the sense of beauty adds a force to the perception of macromolecular structure and function, which will make it possible for you and your students, all of them, to understand macromolecules as they really exist. The TAMS teaching unit was published in 1980 and included sections on the peptide bond, alpha helix, beta structure, tertiary structure, quaternary structure, prosthetic group and active sites. The TAMS unit included 116 stereo pairs of color slides illustrating these sections. A student viewer, Taylor Merchant patented Stereo 7, was provided along with 7 pairs of images for each of these sections, representing a 49 image subset of the full 116 stereo pairs. Each image was accompanied by paragraph of description and a study question. The slide images were constructed using a frame buffer which had 1 byte of memory, 256 colors only for each pixel on a CRT screen. The frame buffer purchased by NIH for making the TAMS images cost $65,000, but a few years later with the more refining, more improvement, it comes only to 12,000, which is on that times respective was a real good price down. Although idiosyncratic color schemes were often used, one of general applicability was developed in collaboration with Bruce Forey and dubbed the functional color code. The hydrophobic would be presented in black color, the positive charge blue color, the negative charge red color, electronegative in peptide bond carbonyl in pink color, electropositive with light blue color, hydrogens same color as atom to which it is attached. They describe the CPK color as C black, N blue, H white, O red, S green, and that was maintained. Still we maintain those functional codes. And then come Evans and Sutherhead's computers. During the 1980s, the most popular computer system for crystallographers was manufactured by Evans and Sutherland. These computers, costing about $250,000 in 1985, displayed the electron density map and enabled an amino acid sequence to be fitted manually into the map. Again, that is a revolution how we could model a protein structure. The color display showed a wireframe rendering of the amino acid chain and could be rotated in real time. These systems use scalable vector graphics. Rapid rotation was accomplished with 3 hardware matrix multipliers, one in each direction x, y and z. The software package often used on E and S computer was Frodo. I will talk about Frodo and its advancement when I talk about the details of a software called Qt. So, Frodo now evolved as Turbo Frodo. Frodo was originally written by T. Alwyn Jones in 1978 and ported to ENS computers by Jones, Ian Tickle and later by many others. A team led by Jones later wrote the program O, popular with crystallographers in the 1990s. Coming to David and Jen Richardson, whom we have talked about earlier, Kine images. During the 1980s, David and Jen Richardson pioneered computer graphic representation of molecular structure with a series of programs developed in the Duke University. In the late 1980s, this led to a program called CHAOS, Chaos, written in Evans and Sutherland's PS300 function net language. In 1992, the Richardson described the Kine image from kinetic image and their supporting program Mage and Prikin. By virtue of its implementation on the Macintosh, this was the first program which brought molecular visualization to a large number of scientists, educators and students. The first time it comes to common scientists. The programs were described in the lead article in the first issue of the journal Protein Science early 1992, and the program itself was provided on a diskette which accompanied that issue. This article also included instruction of using the program Prikin together with Mage for authoring new Kine images. In the subsequent 5 years, over 1,000 Kine images were authored to accompany article in Protein Science. A large portion of these were authored or edited by Jen Richardson. In his own language, Kine images are set up to illustrate a particular idea about a 3-dimensional object. Rather than neutrally displaying that object, they incorporate the author's selection, emphasis and viewpoint. So, this is kind of a revolution, I would say, because when you think about that and when you think about student structural biology, we get a structure and we correlate the structure with function, which is the major challenge where we are working. As I talked about, we have sequence, we have structure and we have function. When you have to correlate between structure and function, you cannot only generate a total structure. You might emphasis on a particular loop, you might talk about one particular active site, one particular amino acid, one particular interesting part, alpha helix, beta sheets, anything, or a interaction between a ligand and an enzyme, between protein DNA, protein RNA, protein small molecule, any sort you want to focus. So, that focusing actually was provided for the first time by the Kinemage. This is at once the great strength of Kinemage and at the same time left a need for a program to allow entirely self directed neutral interactive exploration of any available structure. So, it is good, but it opens up our eye that where the possibilities is going. So, scientists want to do more, and that is where RASMOL was filled. RASMOL was a real good handy tool for us, all of us who have learned around in 1997 to 2002, 3, 5. RASMOL also provided a much richer set of renderings, including space filling rendering complete with options for shadowing and reflective surfaces. So, that time making a figure and correlate it to the function, this was the best home. So, talking about RASMOL, in 1989, while a computer science undergrad student at Imperial College London, Roger Sell became interested in the problem of depth perception for computer representations of solid objects. His goal was to write a shadowing program, a ray tracing algorithm, first enough to allow rotating the shadowed image. He managed to write the second fastest sphere shadowing program in the world. However, at this stage it did little else and it required a specialized parallel processor computer. In 1990, Roger entered graduate school in computer science at the University of Edinburgh, where he continued to develop his program under the mentorship of crystallographer Andrew Coulson. Further improvements in the speed of the shadowing algorithm made inter-processor communication rate limiting on the parallel processor computer. This enabled Roger to implement it on single processor computers such as Unix and later Windows and the Macintosh. Roger developed his program into a much more complete molecular visualization system, and by 1993 it was being used in teaching and for images in research publication. Roger generously made the program available to the world scientific community free of charge when he received his PhD in June 1993. It's a huge contribution, I would say. I still remember I was in my master's in the Department of Biophysics and Molecular Biology in Calcutta University, and Professor Rahul Banerjee from Saha Institute of Nuclear Physics, India used to come, and that was my first love when I see the molecules with the platform of RASMOL. Like me, there are a lot of that time students who was inspired, motivated, and today they are making their career in the field of structural biology thanks to RASMOL. In January 1994, Roger was employed by Glaxo Wellcome, which supported the continued development of RASMOL freeware, including the first version for the Macintosh for the next 2 years. Roger ceased active work on public versions of the RASMOL in 1997, but in 1999 Herbert Bernstein collaborated to produce RASMOL version 2.7. Why RASMOL? The name RASMOL is derived from raster, the array of pixels on a computer screen, molecule. So, raster molecules RASMOL, and Roger specifically mentioned that his initial, which are RAS, is probably coincidental. The fact that Roger generously placed the C language source code for Rasmol in the public domain allowed others to adapt the program to additional types of computers and to incorporate Rasmol's wonderful user interface and rendering into derivative programs. Such derivatives include notably MDL's Chemscape, Chyme and Molecular Simulation Web Lab. Rasmol is very widely used throughout the world. It is difficult to estimate the number of users, but it is likely to exceed 1 million at least. Rasmol is especially popular with students and in locations where resources do not allow the purchase of expensive commercial molecular graphics software, as I have already mentioned my personal experience. Like me, a lot of people have the similar experience. Byran Van Vliet and Tim Maffett at MDL Information System Incorporation spearheaded the development of Chyme Chemical Mime, a visualizer in the form of Netscape Navigator plugin. Chyme uses an adaptation for the rendering and command language from Roger Shell's Rasmol. About 16,000 lines of Shell's source code were converted to C++, made re-entrant and built into Chyme. To this, MDL added more than 80,000 lines of original code to create Chyme version 1.0. Franklin Adler, Gene Holt and others completed the development of Chyme 2.0. Chyme 1.0 was released late in the evening of 12/31/97 and Chyme 2 on November 3, 1998. Chyme was first made available as a beta pre-release in February 1996. Some of the first Chyme based molecular structure presentation to appear on the web were by Henry Repa and David Marcy. The Navigator plus Chyme combination offered many advantages over Rasmol for educational presentation of chemical structure. By early 1997, various authors had developed Chyme based presentations on 30 some molecules and made them available on the web. Subsequently, Chyme interfaces were developed which can load any molecule and apply specialized routines to it or provide a Rasmol like command line permitting any operation of which Chyme is capable on any molecule. So, coming to conclusion of the history, several of the more powerful structure tools with visualization and structure manipulation features are freely available now. Softwares like CN3D, Swiss PDB Viewer, PyMol, VMD, Qt, etc. are coming. As Roger starts with Rasmol, we get a series of mols like PyMol to be like considered as most important, Jmol to be named, Mol Viewer, Mol, a lot of, there was a explosion of different techniques, but if you compare between those techniques, they have two definite category. One category where PyMol, Rasmol and all are there, molecular viewers, they are producing beautiful pictures, beautiful figures. So, we call them molecular viewer, and there are another set where it comes like Frodo, Turbo Frodo, Tom, O, Qt. Here actually you could work on. So, we call them builder. So, one is viewer type of, another is builder type of, and as I told, I am going to talk about the popular softwares. One, I picked up PyMol, which is again a molecular viewer written in Python language, and Qt, which is named on a small bird, is a builder program, and in this current time it is the most popular molecular builder program. So, all those softwares are instrumental in investigating your structures to convey important and useful information for understanding and designing better experiments. Ray trace output is again a revolution. The ray trace output scenes for base rendering and artistic flash have given much better quality to the current publication quality structures. So, if you look at the movie 1 and movie 2, this is a representation of how you could normally work in PyMol, or how you could work and develop high resolution movies. As I told, the current journey is not only restricted to the high quality, high resolution static figures, but we are developing movies which is giving us more insight. So, here is one which is a movie of aquaporin. You see that how the channel is taking part in the flow of water, and this one so this, if I consider these 3 aquaporin, the 4th one is vitipase, which is solved by electron microscopy, taking 1,000,000 million micrographs, and then develop this 3D structure which could move and which shows you the bending, the flexibility, and all the functional aspect of the structure. So, we have gone through a huge journey, starting from the metal made models, going through better models, shifting from there to the Byran bend, and there are many other sculptures which contributed in between. We shifted to computer at last, and then developing better computer model, and current moment we have very high quality movies, very high quality static pictures, which are helping us to understand science in a next level, not only to, as I told, not only to understand or know about a system, but also designing. Protein designing, protein engineering is something which is going through its golden era, and a huge thanks to the molecular visualization. With that, I thank all of you for listening. Thank you very much.